Okay, I lost you there. So first the cell divides into two. And then again, each of those cells divides into two. Can you explain more, please?
Sure, not to worry. Let me give you an outline of the processes occurring during each phase. The centrosomes replicate just before prophase 1 and move to the opposite ends of the nucleus. During the early prophase 1 stage, chromosomes start to become visible as the chromatin coils up, thus becoming shorter and thicker. Also, the nuclear envelope remains intact in the early prophase 1 stage. Then, in the middle of prophase 1 stage, the homologous chromosomes pair up. This process is called synapsis and each pair is called bivalent. The homologous chromosomes condense to form two identical sister chromatids, and each chromatid consists of one DNA molecule. This is followed by an exchange of sections of DNA between non-sister chromatids. This is known as crossing over. In the late prophase 1, the nuclear envelope breaks up and the nucleolus disappears as it does in mitosis. Next, the cells enter metaphase 1, where the nucleolus and nuclear membrane disappear. The centrosomes polarize and help to organize the spindle fibers produced by microtubules. Here, the chromosomes lie on the equator of the cell with the attachment of centromeres to the spindle fibers. Metaphase 1 is followed by anaphase 1, but at this stage, the centromeres do not divide as they do in mitosis. The spindle fibers contract, and as a result, the homologous chromosomes separate and move towards opposite poles of the cell. Finally, in telophase 1, the chromosomes reach the two opposite poles, each with one centrosome. There, the chromosomes start to uncoil themselves and the spindle fibers disappear. Meanwhile, the nucleolus and nuclear envelope reform. In case of animal cells, a constriction appears in the middle of the two new nuclei, dividing the cytoplasm of the parent cell. This stage is called cytokinesis. Eventually, two daughter cells are formed with their own plasma membrane, nucleus, and cytoplasm. As for plant cells, they directly go into the next stage of meiosis without splitting.
Whoa, that was one long process. But it wasn't so hard to understand. Basically, the number of chromosomes is reduced by half during meiosis 1, and in meiosis 2, chromatids separate just like they do in mitosis.
You're right.
Since meiosis 2 is similar to mitosis, I want to try to explain the process of meiosis 2 to see if I remember correctly.
I like your enthusiasm. I have a question for you before you explain that to me.
Bring it on.
Superb, Divash. Now is your time to shine.
Okay, so I'm going to try and tell what occurs in meiosis 2. During prophase 2, chromosomes become shorter and thicker and are clearly visible. The nuclear envelope and nucleolus start to disappear. The centrosomes and centrioles replicate and move to opposite poles of the cell. Then, the cells enter metaphase 2, where the nuclear membrane and nucleolus disappear. Chromosomes lie on the equator, with the centromeres attached to the spindle fibers. In anaphase 2, the chromosome splits at the centromere. The spindle fibers shrink, and the chromatids get pulled apart towards the opposite poles of the cell. Finally, in telophase 2, the chromatids reach the two opposite poles, each with one centrosome. Telophase 2 is followed by cytokinesis. In animal cells, constriction appears in the middle of the two new nuclei, dividing the cytoplasm of the parent cell. As for plant cells, cytokinesis is slightly different. In plant cells, the nuclei aren't separated by plasma membrane yet, so the cell splits 2 times to make 4 cells.